This slideshow accompanies the last chapter on our inheritance section on DNA technologies and genetic engineering. So, in this chapter, it goes over several techniques like DNA sequencing, polymerase chain reaction, DNA fingerprinting, and recombinant DNA. And it goes through these techniques and how they're changing biology and other parts of our society, like medicine, agriculture, and criminal justice. And so we're going to briefly describe these techniques and discuss some of the issues surrounding them and some of the advantages of these techniques. So here I have a little cartoon, and it's a guy in a genetics lab, and the printout there is coming out as a double helix. That's the structure of a DNA. So you see his output paper there. Okay, so this is very technique oriented, and so I'm going to go over the techniques and talk about their use. So the first is DNA sequencing, and if you this DNA sequencing technique uh, begins with DNA polymerase, an enzyme that we can use. It's actually an enzyme made by human cells, bacterial cells, all kinds of cells, and it's involved in copying DNA. And it copies DNA exactly by adding complementary bases. And so these bases are part of nucleotides. And so you add the nucleotides in there and it begins copying the DNA. In addition, you add chain terminators. These are things that end the copying, and they resemble the nucleotides, the same four types of nucleotides, but they're linked to colored dyes. And so there are four colored dyes, and it tells you which nucleotide is at the end of the fragment. So these fragments can then be separated by size, and a computer is used to analyze and organize these fragments. So again, you have source DNA, a single enzyme, DNA polymerase, these nucleotides, and then the chain terminators. And you allow the enzyme to copy pieces of DNA, and the chain terminators end the copy. And this occurs randomly. And so the smallest segment will be the one that ends the first segment, and you can tell how large it is by the color of it and what base is there. And so here it's put on a gel, and the gel allows it to migrate, and the smallest segments migrate the fastest. And so this red one, it corresponds to thymine and tells you thymine is the first. The next smallest fragment is a different color, and that's associated with guanine, and so it tells you guanine is second. So using this, we can sequence whole or partial genomes by doing this, and it's automated. So you just put the thing in the machine, allow it to run for a whole day, and the computer will give you a sequence. And so we can sequence whole genomes of bacteria, plants, animals, or even humans. And so this is a really powerful new technique. This can also be used and combined with other techniques to identify people based on their gene sequences. And this is actually several techniques. We don't sequence the whole genome, but we sequence variable regions called short tandem repeats or frequency length polymorphisms. And so this allows us to use biological samples, hair, blood, saliva, semen, even skin cells, and we can take these samples and we can treat them with enzymes and sequence them, and we get a very exact profile of that allows us to identify someone. There's some links here. I'll put them on the Moodle site, but DNA fingerprinting is really powerful. We used to use things like bite marks and fingerprints and blood types with these samples to identify people. And just for example, there were only 8 blood types. In the whole world, there were only 8 blood types. And so half the people had just one blood type. And so evidence that was based on blood wasn't very good at all. But here we can get things down to individuals. We can identify your exact gene sequences, and so this is very, very exact. This has led to a revolution in the criminal justice system, and um, it's really an amazing impact on our system. We've overturned many, many convictions, and we've been able to convict other people that match the samples exactly who we didn't have any other evidence before this. And so this is, I have a link there to the Innocence Project. There's over 340 overturned convictions and over 150 new convictions. And for example, in Illinois between 1977 and 2000, we executed 12 people for murder. Okay. Once this technique came about in that time period, only the last 10 years, 13 murder convictions, people sitting on death row had their convictions overturned because the evidence that had previously been used to convict them now proved beyond a shadow of a doubt they were innocent. And so these are really powerful new techniques that are revolutionizing criminal justice. Okay, and kind of related to this is when you have a small biological sample like a piece of hair or even tiny cells, how can you get enough material to analyze from that? Well, that same enzyme, DNA polymerase, can be used to copy DNA from a small sample in a test tube. And so we use an enzyme that we've gotten from bacteria from hot springs, Thermus aquaticus. And this bacteria grows in hot springs, and so its enzyme works at temperatures that are almost near boiling. And this allows us to heat and recool DNA to get this enzyme to work. And by repeatedly doing this, we can copy DNA to almost unlimited amounts. And so we can take a extremely small source of DNA, one cell, and we can make unlimited amounts of DNA for analysis. And so this is combined with the DNA sequencing. This is a really powerful technique to allow us to identify and analyze bacteria or DNA from a murder scene, and so really, really small samples. We have more power than ever to identify people based on a single hair or a single skin cell. And there have been some really great examples of the power of this. There was a murder conviction in Texas, a really heinous murder, and there was no eyewitnesses, but at the scene of the crime, they recovered cigarette butts. And so from the cigarette butts, they were able to isolate skin cells, and the DNA that was gotten off these skin cells was copied with a polymerase chain reaction and then analyzed with DNA fingerprinting. And it was linked to DNA in the system of a convicted felon. And with that lead, they were able to construct a case and ultimately convict someone of that crime based on single skin cells. So really, really powerful techniques. Okay. Among the most powerful techniques are a series of techniques that are involved in recombinant DNA, or sometimes called cloning or genetic engineering. And this is when we can take DNA from two or more cells and put them together in ways that they could never come together otherwise. And so we take DNA from a donor cell and we combine it with DNA from a vector, and then we can put it into another cell. And this has been really, really powerful because we can get cells to produce genes. They can produce proteins from genes that they never would have had. So we can insert genes for pest resistance into plants. We can take human proteins and put them into bacteria and get the bacteria to produce medicines like insulin and growth hormone or anti-clotting proteins. This is really all based on the use of a couple enzymes. These enzymes cut DNA in specific points. These are called restriction enzymes. And if you cut two different sources of DNA with restriction enzymes, they will have similar ends, and these ends can be rejoined by another enzyme called DNA ligase. And so you can put together DNA from two different sources. This allows you to put DNA into a plasmid, which is a piece of bacteria or DNA from bacteria, and it's easily transferred from one cell to another. So once you've put DNA into a plasmid, you can easily put that into a new cell. Okay, this has allowed us to make insulin from a human gene, and we've been able to insert it into a bacteria. So the bacteria produces that gene, and we can easily harvest that. We make insulin that way, growth hormone, erythropoietin for producing blood, anti-clotting proteins, clotting proteins. We make vaccines that way. We can also take genes for resistance to crops, particularly a toxin made by Bacillus thuringiensis. And we've taken the gene out of that and inserted into the corn. This produces a compound that's toxic to corn borers, but is harmless to people. And so, in fact, corn produces its own insecticide. Okay, virtually all the corn produced in the United States carries this gene now, Bt corn. We also use it to produce crops that are resistant to herbicides, crops that have improved nutritional value. We use this to make enzymes for industry, all sorts of applications worth billions and billions of dollars. In addition, we can take DNA and use it to treat illness in a living person by giving them genes that maybe can replace their own damaged or missing genes. The potential for this is to treat cystic fibrosis, muscular dystrophy, hemophilia, type 1 diabetes. All these disorders involve a missing gene or cells missing genes. And if we know the right gene, we're developing the ability to insert it into cells. This is proven more difficult than was originally thought, but new techniques have helped us improve this. And so there's a potential to treat genetic disorders by giving people the correct gene. Oh, okay, so really vast improvements, a revolution in our ability to manipulate genes and to transfer genes. This is a rapidly advancing field, a field that's advancing so rapidly that sometimes these advances scare people. We're able to make organisms and do a form of breeding that we could never do. We can test and identify people from extremely small samples, and we can find out things about them that they didn't know themselves. This plays on people's fear. People are worried that if I eat genetically modified food, maybe that gene is going to get into me, or maybe that gene will just spread in the environment and become a superorganism, and corn will just grow everywhere. Or maybe we will genetically modify people so that the genetic modifications of people will replace normal people. I think many of these fears are overblown. The transfer of genes can only occur in the lab under very specific conditions, and we can only transfer one gene at a time. And so we can't transfer whole groups of genes, and we can't transfer genes that affect really complex characteristics. That's pretty far beyond our ability. And so the idea that you're going to eat a food that will genetically transform you, that's just a mistaken idea and is not true at all. And there's no way we're going to create hybrids between fish and people, or between horses and people, or anything like that. But there are ethical issues involved in biotechnology. Some cases people may be injured, but other issues are more mundane, like who owns DNA? Just because someone isolated it, it doesn't mean they invented it. There's a big difference. But perhaps if the patents can be granted on a gene, maybe somebody owns the genes that you're using right now. So who owns DNA, who benefits, and who decides are big questions. And the technology can invade people's privacy. And so there's examples of cases where companies have tried to identify someone doing something they didn't like, and they've taken samples from their employees or forced their employees to give them samples um, that not only can help them identify things, but maybe decide on whether they should insure this person or not. In other cases, people have patented genes, like the breast cancer gene, BRCA1. This is a gene that wasn't invented by a researcher, but it was discovered. And because it conveys a risk of breast and other cancers, they patented it and patented the test. Um, well, now the test is very expensive. Uh, how long does that patent last? If if that high price of the test discourages people from getting tested, it might be that their discovery actually makes people's health worse than if they wouldn't have patented. What if employers do this test to decide who they want to hire because those people that have the gene might have higher insurance costs? And so when I think of ethical issues, more of them are more mundane than some catastrophe, but they're involved in democracy and fairness and justice. Okay, I'll post some links underneath here for some really good videos that you can see. Check them out and respond to the forum, and we'll talk about the dates for testing next week. Thanks.